Well, welcome everybody. Um, my name's Jeff Miller. I'm the founder and CSO of In Vivo Scribe. And what's really fun to see is I see some faces in the audience that go back to when we started our company 30 years ago at this conference. So it's really, I realize some people are getting ready to retire, but we've had a good journey, and I think we're leaving this in good hands. And we're not done yet, so we're just getting started. So we're very excited to see you. So we're fortunate today to have really two great speakers. And we have Dr. Nitin Agarwal and Kristen Floyd from MD Anderson. Dr. Agarwal is a computational scientist working at MD Anderson in the molecular diagnostic lab. Bachelor's of Science and Master's in Pharmacology and Drug Assay Development. And then his PhD in Biochemistry Pharmacology from the Georg August University in Germany. Over the past decade, also he did a postdoctoral fellowship at MD Anderson. So over the past decade, he's developed a number of different innovative molecular diagnostic tests, and he's going to tell us about the journey with FLT3-ITD and NPM1 here today. Kristen Floyd is a laboratory manager at MD Anderson, so she keeps everything rolling. And she's a graduate of the University of Georgia, go Dogs, right? And so she manages, as you know, the people who manage the lab really completely help us get all the results and are instrumental in doing everything. They're the backbone of our work here. So she's developed a number of different CLIA cap assays. She's also contributes in process improvements and making sure the latest biomarkers are coming up in the lab in a timely way, and implementation of cutting edge technologies, which we all have to stay on top of as this field grows and becomes more mature and better in precision medicine. So, as I said, we're very fortunate to have these two speakers. So, Dr. Agarwal.
Thank you, Jeff, for the kind information. So I just want to add a little bit more about my CV. I am a data computational scientist, but I am also developing assays in MD Anderson. So this assay, FLT3 and NPM1 assay, has been developed with my hands. I did hand-on experience. And with our MDL bioinformatics team, we are able to successfully validate these assays based on the CAP guidelines, and now both the assays are running successfully in MD Anderson Cancer Center. So before I start my presentation or talk, I would like to disclose I and Kristen both are full-time MD Anderson employee. We are receiving honoria, and we have used In Vivo Scribe and FLT3 and NPM1 reagents with In Vivo Scribe. Outline of this talk will be, so I will talk about a little bit background about MRD and its diagnostic importance, why diagnostic importance is necessary in both these cases. I will also discuss or share my experience for the validation of these assays in MD Anderson, and I will show some results, how it's going to come. Kristen will show or share her experience with the clinical performance in MD Anderson in this laboratory. So what is AML is already known for clonal proliferation, and based on initial mutation and gene fusion, this process progress. And once it moves forward for the progression of AML with the help of NPM1 mutation and other genes, and finally FLT3 hit it and it fully progressed AML. So based on these mutations and fusions, AML has been classified 3 categories. It's favorable, immediate, adverse. I am not going to go for detail. I assume that everything is already known. But I would like to note it that both FLT3 and NPM1, both are coming in the intermediate stages. Once the subject has been mutated, once the subject has been finalized, mutation and fusions are there, a frontline FDA treatment is going through, and some subjects are going for intensive therapy, some are not. Finally, or eventually, subjects are getting complete remission, achieve complete remission. The most important is when the subjects are received or achieved complete remissions, then some of the dormant tumor cells exist there, and later, as the time passes, it progresses, and once again, tumor arises. So it is very, very important and critical to diagnose these MRD cells. And this depends upon how the assay sensitivity is going through it. So next question arises, how the MRD can be measured? We have some methods already known, and they are going through. First is multi-parameter flow cytometry. This method has been applicable, or is still applicable in a lot of labs. The disadvantage of this method is it required expertise, but it is not as sensitive. Next is qPCR. This is highly sensitive assay. It has only one limitation, that it is one analyte, one assay. We can use this qPCR only for one gene. As compared to NGS, NGS is a newly developed assay, so it is as sensitive as qPCR. But in some cases, if the tumor cells are evolved after mutations or after MRD, so we are able to detect those cases also. For example, this is a slide where we have a complete remissions patients. We have two bars. One is yellow bar, one is blue bar. Yellow bars has some MRD positivity, whereas blue bars has MRD negativity based on qPCR assays. And you can say that in the blue bars or blue line, we have a very, very less cumulative incidence of relapse, as well as overall survival is much, much better when the MRD is negativity. The same overall survival and disease-free survivals are much, much superior in MRD negative case. So all these cases, what is shown us that detection of MRD is very, very important to be a better subject care. Now, I am going to, because both these assays are running successfully in MD Anderson Cancer Laboratories. So we are using NPM1 assay for the baseline as well as MRD tracking. We are using FLT3-ITD assays also for baseline as well as MRD tracking. Later, we will continuously go for MRD tracking. We have developed our own demultiplexing pipeline, which Kristen will share in later slides. And we are using in vivo Scribe software for analysis, and we have our own report format, which she is going to explain you later. So in my experience, in last 2 or 3 year experience, I never saw such a simple library preparation for NGS assays. It's a 2-step process. So you have to extract your DNA, mix with the master mix, which is provided by the in vivo Scribe, 2-hour PCR reaction, clean up with the beads, your library preparation is done. So you have to quantify your library and pooled and sequencing. Overall, this whole process, it will take 70 hours. And most of the time is going to the sequencing, because we are using MySeq sequencing, paired 300 cycles. It will take around 56 to 60 hours for whole process. Another important thing is you can mix both NPM1 and FLT3 samples in one sequencing workflow so that you can save the time if you don't have enough samples for that. And that can be done in this step. So after PCR, you have a different indexes. You can mix and pool all the NPM1 and FLT3 sample together and do the sequencing. After demultiplexing, both the samples have been segregated, and we can do the analysis separately in vivo scratch software. Sorry? 20, we have developed assays between 15 to 24. So it depends the volume what you have it. You can go lower also, but it may be the expensive for you. So the most important assay for the development or for the validation is the linearity and the LOD. So linearity, which we checked it between 0.002% to 49%, and you can see in case of NPM1, we have a very good linearity. When you see the R-square value is 1. We used OCI AML cell, which is already known for type A NPM1 mutation. Similarity, or in similar way, we use FLT3-ITD also with the different range. And we use MV411 cell line, and we got a very strong and good linearity. Based on these results, we selected 0.005%, and we go for 20 replicates for both the assays. For NPM1, you can see the raw data with our hand that all the 20 cases, we have enough number of reads to detect or to say positive results. And we have set the criteria 5 or more reads for that case. Even we can go a little bit lower also, but we just go through and go for that one. The same results we are getting with FLT3 and ITD also. You can see that all the 20 replicates has passed with enough number of reads. Next, we go for accuracy. In accuracy, in case of NPM1 mutation, we used 58 subjects. Out of 58 subjects, we have 52 positive, 6 negative. In 52 positive, we have all type of NPM1 variant from A to Z and others. I will show you the example in next slide. And you can see that positive PPV and PV value is getting 100%, and we are getting 100% sensitivity as well as specificity. This is the example what we got, and we selected the sample, and you can see that in the yellow color bar, we have a reference and leukemia. This is our panel. We are using Haloplex NGS assay, where we have detected this percentage of variant as well as type of mutation. On the green bar, you can see that this is MRD, NPM1 MRD assay. We accurately detect all type of variants, and it is 100% concordant. The only difference what we found is that in MRD, ABD, and all the other variants, what they are described in the form of other, as compared to our and leukemia, we have a separate one. Then we go for accuracy for FLT3-ITD. We used 64 subjects. Out of 64, we have 54 positive. Please note in this 54 positive, we have a reference method is gene scan. In this gene scan, we have 1 ITD, 2 ITD, and multiple 3 ITD. So we select all type of ITDs in this 54 subjects. We have a 10 negative, so overall, we have 100% PPV, 100% NPV, and 100% sensitivity and specificity. So in case of FLT3-ITD, we can detect 9 base pair to 252 base pair insert For examples, you can see there are two examples we are showing, one for 42 base pair inserts and one for 200 base pair. When we compare with the gene scan, the most important thing what we found it, when they have one ITD, we are getting one ITD with the MRD assays. But when you compare 3 ITD with the gene scan, we are getting multiple ITDs in MRD. So that itself is showing that those cases have a destabilized FLT3-ITD. So a lot of cases, we are not detecting some ITD which is already present in patients. Next, because we are working on 0.005% sensitivity, we need to detect whether there is any carrier crossover contaminations are available or not. So to perform this experiment, we have a checkerboard panel. So we use one highly positive, negative, highly positive, negative. So based on that checkerboard, we prepare the library and do the tape station. And you can see that all the negative, all those alternatives, they are showing the negative. You can see the accumulated average concentration of NTC, which is coming 195 picomolar. And we have set the limit for equal to or less than 1,000 picomolar. Additionally, we also spiking some of the highest concentration NTC. For sequencing, we are unable to get enough number of reads so that we can detect the variants. This itself is showing that we don't have any carryover and cross-contamination. Same thing, we perform FLT3 also, same checkerboard, and we are getting the excellent results and showing that both these assays are very robust and they don't have any carryover and crossover contamination. Next, we go for interference substances. So we have two types of interference substances. One is specimen specific, and another one is method specific. Specimen specific, we already have a very good quality of DNA, so we are not expecting anything with the DNA quality, whereas method specific, the most critical step is cleanup beads. In lot of cases, when the person is doing the cleanup beads, they have little bit remnants of the beads and ethanols exist there. So we want to see whether after adding 5% beads or 5% ethanol, if it is there, it's affecting the results or not. So we have a 3 group. One is the neat one, where we don't have anything. And the second group is 5% impure beads. At the final step of cleaning bead, we add 5% impure beads. And the third group, we have 5%, 80% ethanol. At the end, before we go for tape station, we add 5%, 80% ethanol. And then we did the sequencing, and we found all the results are concordant with the neat one. Same results what we are getting in the same both interference substances, 5% impure beads, as well as 5%, 80% ethanol. We used it as an interference substance in FLT3-ITD assays. And we found that these are not affecting any results even after adding 5% beads are there. The most important experiment is the specimen stability, where we took the specimen, keep it for 7 days and 21 days, and then extracted the DNA. And after extracting the DNA, we want to compare with the day first and see whether we are able to see the results or not. You can see in case of NPM1, when we compare with the day one is a specimen when we just collect it, and within 24 hours, we extracted the DNA and analyzed it. Day 7, when we keep the specimen for 7 days in refrigerator, extract the DNA and analyze the results, you can see that all results are concordant, and we have a very good linearity. In case of FL, somehow we don't have a 21-day samples because it's a bone marrow. We don't have enough bone marrow. So I am unable to do the experiment in case of NPM1. But FLT3, I have a 21 days few samples. And when we compare with day 1 and day 27 or 21, we are getting the same results. So all these results are showing that this assay is very robust. Even if the DNA is degraded, it is not affecting the overall results. To conclude, the summary of this assay has a very good linearity. It's a big range. It can detect up to 0.005% and little bit lower also. It is highly specific, more than 5 reads, has a positive accurate values. LOD is going to be 0.005%. And the fourth one is they have a very robust, highly reproducible and specific assay.
So Dr. Agarwal did a great job of discussing how we've validated the FLT3 and NPM1 MRD assays. So I'm going to kind of touch on how we've incorporated those into our test menu. So we do have an NGS-based panel that we use at baseline for AML patients for NPM1 and FLT3-ITD detection with a sensitivity of allelic frequency down to 5%. One of the limitations here is if the FLT3-ITD is too large, the NGS methodology that we currently use cannot detect it. So the gold standard for FLT3 baseline ITD and TKD detection is the gene scan or capillary electrophoresis. In terms of in the laboratory space, this is very quick and cost effective. We can get results within 48 hours with a sensitivity down to 1%. And then for our follow-up samples, it's been crucial to use the MRD NGS assays that have an allelic sensitivity that we were able to validate down to 5 times 10 to the minus fifth, or 0.005% VAF. So for our follow-up AML subjects, we do incorporate a testing algorithm. At baseline, we would perform an NGS panel. Even though it does have a lower sensitivity, it may give us more information. Once that NGS is performed and we know that whether or not the subject has a FLT3-ITD that's detected, or if it's not detected, we would then follow this testing algorithm. If the ITD is detected, we would then follow with the in vivo scribe MRD assay to follow the clone. And we would not order the comprehensive NGS panel. If the ITD is not detected at baseline, we would continue with conventional FLT3 gene scan testing for the emergence of a new clone, or potentially to follow a TKD. And then we would continue to order our in leukemia panel for other known MRD markers. So Nitin kind of went over the workflow a little bit, but we'll dive into this a little bit deeper here. As I mentioned, we kind of triage and ensure that we're incorporating the appropriate tests to ensure that we get the most information. For the FLT3-ITD MRD workflow, we do pair the baseline with the follow-up. One of the limitations of the gene scan is that you're only going to get the size of the ITD, you're not going to get the sequence. So we do sequence the baseline to ensure that the baseline and the follow-up correlate. After we've triaged, we know the appropriate test that we're going to order. We ensure that we have the minimum concentration. With the allelic sensitivity that we've established, the assay does require 700 nanograms of input. Once that's established, we would begin with the library prep. As Nitin mentioned, this is a very user-friendly assay. It's one PCR followed by a cleanup, followed by quantitation. We use the tape station to quantify the libraries. We do pool and sequence both the FLT3 and the NPM1 samples on the same flow cell, and that does help with reducing cost and associated turnaround time, as previously mentioned. Another assay that you could potentially couple and sequence with is a great feature that in vivo scribes assays. Their indexes are compatible with other kits, so we also could sequence the IGHV in addition to the NPM1 and the FLT3. We would sequence on the Illumina MiSeq. We have established a robust quality control, and so after sequencing is completed, we ensure that all of the QC metrics, the number of reads that are required for the limited detection that we've validated, ensure that that's present, and then we would continue with our bioinformatics pipeline for demultiplexing and alignment. We would then parse in our in-house developed software, OncoSeq. So this software serves as a way to view the results from the annotated software, as well as a database, so that we can track and trend the results over time. Once it's parsed in OncoSeq, we would then do a positive and negative control sample correlation, correlate the samples with cliniopathic findings, correlate the baseline to the follow-up, and then issue a report. So this kind of outlines the custom MD Anderson bioinformatics pipeline. Post-sequencing, we ensure that the sample sheet is appropriately annotated for the sample or the test that was ordered for the sample, whether it be FLT3 or NPM1. We would use an in-house software to perform the demultiplexing, the alignment, and QC, followed by the Dockerized FLT3 and NPM1 MRD in vivo scribe software for further annotation, and then we would parse in OncoSeq for review and report generation. Here's a look at what the results would look like after we parse it in OncoSeq. Of note here, it does give you the read depth to ensure that you have the minimum amount of reads for the level of sensitivity that you're looking for. It pulls in whether or not that the ITD result is positive or not detected. It gives you the ITD size, the reads, as well as the variant allele frequency. And here's a look at a negative sample where it also gives the read depth and then an ITD result of not detected with a high confidence, close to 100%. So here's a use case of a sample. We incorporated the minimum 700 nanogram DNA input with a read depth of greater than 1 million reads to achieve our limit of detection of 5 times 10 to the minus fifth. And we were able to detect a 4 base pair NPM1 duplication at 10 times 10 to the minus fifth. And then this is a use case where it's comparing the conventional method, the gene scan, to the MRD assay. You can see here that this patient or this subject had multiple ITDs detected by the capillary electrophoresis, as seen by the multiple peaks at the bottom. And then with the MRD assay, you're able to have the sequence associated with each of the ITDs for follow-up and tracking. This is an example of a report where we're able to report the sequence and the associated VAF. So in summary, the FLT3-ITD and NPM1 MRD assays have an allelic sensitivity of 5 times 10 to the minus fifth mutant alleles per total alleles. You can easily multiplex the samples and targets in one sequencing run. It utilizes a Dockerized software that's very user-friendly and can be coupled with existing bioinformatics pipelines within your laboratory. It's a standardized workflow with minimal user error, and the NPM1 MRD assay can detect insertions greater than 4 base pairs. The analytical sensitivity of the assay was validated for an ITD length of greater than 9 base pairs, while the maximum ITD length detectable by the assay is 252 base pairs. The detectable size and sensitivity vary depending on the insertion location and sequence of the ITD. And then lastly, additional variables such as sample quality Hemodilution may affect the ability to detect and determine variant read frequency, and correlation with cliniopathic features are recommended for a complete evaluation. And we'd just like to acknowledge our medical and CLIA lab director, Dr. Patel, as well as some of our faculty, Dr. Lagavi, Dr. Rockport, and some of our data scientists, Wei Chen, Shibu, and a vast majority of our team at the Molecular Diagnostics Laboratory.